机会让外行人来参加这个 expert meeting，觉得很荣幸。Today is my great pleasure to attend this expert meeting, and here I would like to introduce our true expert, real expert in urological oncology, the Professor Hussain. Professor Hussain is the chair in the Professor of Consultant in Medical Oncology of Institute of Translational Stratified Medicine of Plymouth University and the Peninsula School of Medicine and Dentistry. And if you have seen CV, you will know he is the PI of several major important pivotal study of the urothelial carcinoma. So I think there is our great pleasure to invite Professor Hussain to discuss with us, and we are also very glad to share the IO experience in urological oncology with Professor Hussain. We are looking forward to a fruitful discussion with the expert meeting. So let's welcome Professor Hussain.
Very much for the wonderful and introduction. It's a great pleasure for me to be here and to go through the journey of metastatic bladder cancer. We all share the same issues and feelings where patients with advanced bladder cancer quite often given a Cinderella status, and while research has been ongoing, but at the same time, over the last 35 years, very little change in bladder cancer. But over the last 5 to 10 years, there have been significant changes in bladder cancer, and the landscape has significantly changed. So I will share with you all the enthusiasm about bladder cancer and management today. And we will then talk about some of the reimbursement and nice sort of a pathway, how it falls, knowing that you guys will be going to the reimbursement process for atezolizumab in Taiwan as well. So, in terms of, as I said, over 30 years, nothing changed really. And the metastatic bladder cancer, for example, it's cisplatin eligible first line, median survival remained around 14 to 15 months. It didn't change a lot. For cisplatin ineligible patient group, the median survival was about 8 and 9 months. And that is why I call that this is an unmet need in terms of advanced bladder cancer, where patients have not significantly, in terms of there's not been significant change in outcome. And that is where we have got a huge potential opportunity of having a big impact on our patients. So this is from the Maria DeSantis work, 9 months versus 8 months from big clinical trials, showing that median survival in cisplatin ineligible patient group is quite poor. This is some of my own work looking at split dose cisplatinum with gemcitabine for patients with poor renal function that we previously published as well. And it is used in many centers, but this is a small phase 2 trial which showed that renal function remained stable with a good survival benefit as well in terms of median survival. Where things have significantly changed and made a huge impact is looking at this sort of immunotherapy, which has literally transformed bladder cancer. Again, today there is limited time. I think we're talking more about it tomorrow as well. But we'll talk about informal discussion that I've got a number of patients whom I've treated with atezolizumab in InVivo 210 and 211 studies, where patients, for example, InVivo 210 patient who 4 years down the line is still with us with metastatic bladder cancer and his disease remains very stable when we stopped atezolizumab after 18 months of treatment because of colitis. So those patients are not anecdotes. We are getting more and more patients like that who are alive for prolonged periods of time. So immune checkpoint inhibitors play a key role. There's multiple steps that are involved, which many of you are very well involved and used to. In terms of the first PD-L1 inhibitor, if you look at in terms of survival benefit, we can see significant survival benefit in cisplatin ineligible patient groups with the survival of 15.9 months. If you remember the key figure of 8 months or 9 months in the cisplatin ineligible patient group, this is almost doubling of survival benefit, and we were all excited when we saw that, because that was a new step forward towards advanced bladder cancer for cisplatin ineligible patient groups as well. With pembrolizumab as well, we saw a very good response rate, and more recent data is showing a median survival of 11 months with pembrolizumab as well in cisplatin ineligible patient group. This is sort of how pembrolizumab works, working through inhibiting binding of PD-L1, PD-1 and B7-1, and then in that sense enhancing the T-cell priming and reinvigorating suppressed immune cells, and leaving the PD-L2, PD-1 interaction intact, and in that way may sort of preserve the peripheral homeostasis, so using the positive effects in terms of controlling the disease. Focusing on platinum ineligible patient group first, cohort 1, which was 119 patients, cisplatin ineligible, atezolizumab 1200 milligrams again, and the cohort 2, which we'll talk later on, platinum treated metastatic urothelial cancer. Very clearly, what we saw was that in terms of IC 1 to 3, 24% response, 28% for IC2-3, and in terms of all patients, it was 23%. The change in tumor burden was quite interesting, that we were continuing to see significant change in tumor burden, improvement in outcomes in view of that, and the durability of response was another thing which was quite interesting to observe, that patients have been responding and responding for quite a prolonged duration of time. That durability of response is the key thing. When a patient comes to our clinics, they're not very interested in telling them the median survival of 8 months or 12 months or 14 months. What they're very interested in is that there's 1 in 3 chance that these patients will be alive at 12 months, 18 months, and 2 years down the line. And those are the key figures that the patient asks us. Will I be seeing the next Christmas? Will I be seeing a Christmas after? Based on that data, we can tell them that actually there is a 1 in 3 chance that patients will be alive at 18 months and further on. So those are sort of key things which literally help NICE to make a decision that based on that we should be allowing these drugs for our patients. This is the overall survival by PD-L1 status, and again you can see that overall 15.9 months. What is paradoxical here is that actually IC0 and 1, which is quite a decent number, 87, the median survival is 19 months, versus IC2-3, which is 12 months. It's still significantly more than 8 or 9 months that you see with chemotherapy. Treatment-related adverse events were sort of much better compared to chemotherapy that we see. No neutropenic issues. There are immune-mediated toxicities that we as a team has to be aware of, and that my industry colleagues have to be very clearly focusing on as well. As more and more clinicians start to use these immune checkpoint inhibitors, it is very important that industries play a very key role in making sure that the patient's toxicity are very well managed, and the clinicians are sort of properly trained on that, because some of these immune-mediated toxicities could be quite difficult in a general hospital. It's very easy to miss it. Even in our centers, sometimes you do see that patients are left where they are significantly fatigued and unwell, and when you do the ward round, they say, oh, this patient is on third-line treatment and we thought he's dying, while you know that this patient has immune toxicity, endocrine toxicity, and you start them on steroids, and the patient is back to your clinic next week, who was being left thinking that, oh, it's third-line metastatic, must be progressing and he's dying. So those sort of things will happen, and I'm just warning you to make sure that you keep an eye on these things, and that's where industries in UK have played a very key role in developing educational sessions to help improve on the outcome for these as well. And that helps to improve the access for patients, because when you see good responses, you carry on treating them, and where centers which have treated just 1 or 2 patients and they are uncomfortable because they saw some toxicities that they were very worried about, and those centers never use the drug again. So it helps the patient, it helps the access of these drugs to the patient, and it helps the continuity of treatment for the patient if we can manage these toxicities in a much better way. So these are some of the toxicities that are treatment-related AEs, and then immune-mediated toxicities as well are important to look at. For example, rash, ALT increase, again, it is 1 or 2%, and that in itself is important to remember. Because of the small percentage of patients who are getting toxicity, it's quite easy to miss it. If we are not looking specifically for liver toxicity or renal toxicity, we can easily miss these sort of things. So those are sort of things that we will need to be keeping an eye on. And use of steroid doesn't lead to poorer outcomes. So this is with the 052 studies. Again, very similar design, looking at pembrolizumab in platinum ineligible patient group to look at that. And then similar sort of criteria, looking at patients who are cisplatin ineligible by definition, looking at the endpoints, objective response rates, and then PD-L1 expression status. And then PD-L1 status assessment was done in a different way, looking at a combined positive score compared to looking at tumor immune cell infiltration. And these are sort of things that we will talk about hopefully in our discussions in more detail, that these different ways of assessing the status is quite difficult to interpret. Similarly, we need to be aware of that PD-L1 is a dynamic biomarker that can change with time with treatments as well. And that's why multiple biopsies perhaps might help us to see how PD-L1 status actually changes. Results: 541 patients were screened, 374 were enrolled. Sort of what was quite clearly the characteristics and things were quite what you would expect in advanced metastatic bladder cancer setting. Nothing but key things that duration of response, again with PD-1 inhibitor, pembrolizumab, we continue to see, just like PD-L1, good response as well. And in terms of the overall survival, the median overall survival was 11.5 months. So it's a little different to what we saw with atezolizumab, which was 15.9 months median overall survival. But again, it's still very significant, very important compared to 8 months or 9 months with chemotherapy. And in terms of the toxicity profile, there was no major issue. So what we are seeing with immune checkpoint inhibitors, consistently good efficacy with toxicity, which is very well managed, as long as we are carefully looking at some of these toxicities like hypothyroidism and hyperthyroidism that can occur with immune checkpoint inhibitors as well. So in terms of toxicity, again, adverse toxicity, the key thing, as you will see, the percentage of patients who are getting grade 3 or 4 toxicities are very, very small. And again, that is one of the major reasons where some of these toxicities like hepatitis and things can be quite easily missed, where somebody with liver mets can be, one can assume that it's liver metastasis that is getting worse, but it could well be completely related to the drug. So we will need to be very carefully watching that. So with 11.5 months follow-up, again, median survival of 11.5 months was quite significant in terms of platinum ineligible patient groups. In terms of immune checkpoint in platinum refractory setting, it is what we are seeing is multiple drugs are there. They're being used, atezolizumab, nivolumab, pembrolizumab, amilimumab, durvalumab, good response rates, and in terms of survival as well, we are seeing significant survival benefits. PD-L1 expression on immune cells was evaluated with Ventana, and again, there's been a lot of debates around that, a lot of criticisms around that. Who is PD-L positive, who is PD-L negative, and how do we assess it, and how do we move forward from it? I think what I can tell you is that behind the scene, a lot of industries are now working together actually, which is a great positive sign, to try and define it in a better way to see actually how do we define PD-L positivity versus negativity. So that's a very welcome thing that is ongoing. So if we go to overall survival, you can see median survival was 7.9 months. But I think more than, as I said, it's a big phase 2 trial, 310 patients. What is key here is not the median survival, but the durability of response in the subset of patients. How can we develop predictive biomarkers to find out? That is an ongoing thing, which I will try to touch at the end with few slides, unless we start to run over time. And it will tell me that I should not go into much more detail. Then we can talk about it later on after the meeting. But key thing is that predictive biomarkers are being developed, CD8 infiltration, tumor mutational burden, things are looked at, and the PD-L1 expression. So they are complementary to each other and they are hypothesis generating. But I can tell you that huge amount of effort is being done to try and look at the predictive biomarkers. So median overall survival is not the key thing, but it's the durability of response and the percentage of patients who are living at 12 months and 90 and sort of 18 months down the line. Treatment adverse events, no major concerns, no major issues really in those 310 patients. Similar efficacy that we have seen, but in terms of toxicity, much better tolerated than chemotherapy. Immune-mediated adverse events were seen, but again, small percentage, pneumonitis of 1%, and that is why it is important to look at these things, treat them aggressively with steroids, and that can have a big impact on continued treatment for these patients. So those are sort of things that we as a team has to work together to try and improve on the outcome for our patients. This is the Indigo 211, which we published in Lancet just a while ago. It's a big study. It's one of the largest randomized metastatic bladder cancer trials, and that is one of the most important things that I would say, that this is a big experience in metastatic bladder cancer. And again, the trial was designed to look at the hierarchical testing, looking at PD-L1 positive patients versus PD-L1 negative, which was supposed to be the strength of the trial, but unfortunately, because of the PD-L1 biomarker, the way it behaved, that made the trial to be negative. And that we will discuss today, now, and perhaps later on at the end of the meeting as well in informal discussions, that what were the positives. As I said, one of the most important thing is the largest randomized trial of metastatic bladder cancer with 931 patients. So chemotherapy versus atezolizumab was done, vinflunine, docetaxel, or paclitaxel, so very sort of a very good design, very pragmatic, that completed recruitment within 1 year. 77% of the patients came from Europe, who was used to vinflunine, and that's why perhaps half of the patients on chemotherapy got vinflunine compared to the other half who got docetaxel or paclitaxel. And the reason I'm mentioning, because these are some of the differences between this trial and the pembrolizumab trial as well. So the trial looked at this 1363, 931 enrolled, and sort of well balanced in that sense. What was key, again, in terms of the factors, there were no significant differences, and I'm sure you have looked through these things. In terms of the patient groups, there was not much difference. The outcome, IC2-3 patient population, there was no difference in that sense. What it told us that PD-L1 was not selecting patients for response, but actually it was irrespective of whether you treated them with PD-L1 inhibitor or chemotherapy, the patient did very well. So it turned out to be more like a prognostic biomarker, where if you are PD-L positive, you will do well, whether you get immunotherapy or chemotherapy. So that was one thing which happened with this trial. And 11.1 months versus 10.6 months. So that made this trial to be, by definition, by primary endpoint, the trial to be called negative. But there were lots of positives within that negative. For example, if you look at intention to treat analysis, just like if you look at the 045 study, the pembrolizumab study that was done in second-line setting, intention to treat analysis, overall survival, then you see a hazard ratio of 0.85 with a p-value of 0.038. So that all of a sudden statistically becomes significant. And again, 39% of the patients alive. So you treat 5 patients and 2 of them will be alive at 12 months. And that 12 months, then durability, you see that ongoing durability, which is quite key as well. Response by PD-L1 subgroups, again, you can see durability of response was seen in intention to treat population, and 63% of patients in the atezolizumab arm and 21% in the chemotherapy arm had ongoing responses. So there's a significant improvement in long-term durability of responses. Treatment-wise, big data set tells you that toxicity actually is much better, so quality of life-wise, things are much better with atezolizumab compared to chemotherapy, and significant difference between chemotherapy and atezolizumab, which again was one thing which NICE looked at it and was quite impressed with the toxicity profile of this drug, to say that compared to chemotherapy, less admissions to the hospital and less toxicities. The primary endpoint was not met, but there was no safety issues, and then there were different significant benefits that we saw in terms of durability of responses as well, and the percentage of patients who were living 12 months, 18 months and down the line as well. So in terms of overall response rates and durability of response were consistent with the 210 data, and the 12 months were significant, were similar to what we saw from the phase 2 data. You're seeing 39%, 37%, so it's very similar to what you saw in the Indigo 210 phase 2 data, and the durability of responses was seen as well. So was the study negative? Yes, the primary endpoint of overall survival was not met. So it did not meet its primary endpoint for improved overall survival in patients greater than 5% PD-L1. And that is key to remember, that this is negative because of those 200 patients. Overall, if you look at 931 patients, then intention to treat analysis, you have a statistically significant p-value. So statistical design was challenging, stepwise hierarchical testing. Chemotherapy control arm exceeded protocol design assumptions, and again, as I said, vinflunine played a key role there. That vinflunine arm did much better compared to the pembrolizumab arm, where the vinflunine arm did worse. And again, there are some imbalances. If you look at, there were more patients with liver metastasis, almost 47% of patients with liver metastasis who got vinflunine in the pembrolizumab trial versus chemotherapy. So those are certain things which were a little bit unbalanced. Chemotherapy control arm exceeded protocol design, and PD-L1 expression did not behave as anticipated. So these were the key issues that happened, which perhaps explained why it didn't. But at the same time, PD-L1 as a predictor, what we saw, IC2-3, it was initially coming out that IC2-3 will do much better, but in terms of the cohort phase 2, you can see IC2-3 again doing better, but overall what is happening is that in terms of the subgroups, for example, in the cisplatin ineligible, there was not much difference as well. So there is contradictory things that are going on, and that is perhaps one thing that we learned from it, that it is a dynamic biomarker which may be changing its sort of status as we go along as well. So yes, the positive among the trial was sample size, consistency in efficacy data, consistency in safety data. So these are sort of key things that have really moved the field forward in terms of understanding these settings. This is a updated analysis of the Keynote 445 with pembrolizumab, which again very clearly showed similar design, looking at 200 milligrams of pembro versus the chemotherapy. There are some differences. Again, the assessment was different. It was a combined positive score, looking at tumor cells and immune cell infiltration. But that was something I was mentioning about pembro versus chemo. If you look at the liver mets, it is 1 in 3, 33% between pembro and chemo, which is exactly well balanced. But if you look between chemotherapy, there is a difference. Almost 50% of patients with vinflunine had liver met. And remember, it was vinflunine that actually changed the impact in the INVIGO 211. So again, these are sort of things that one needs to look at. It's not, the trial has been designed and has been reported very correctly because it is looking at chemotherapy versus pembrolizumab. But at the same time, there are some inherent differences which may explain us the differences that we saw in terms of outcome, in terms of median survival benefits, because in the pembrolizumab 045 study, it's the vinflunine arm that does the worst. And perhaps that's one of the major reasons. And then you also look at the performance status 2. So performance status 2 is much heavier. 1 in 3 patients with PS2 were in vinflunine arm compared to 1 in 4 in every other arm. So there are certain differences in performance status, and liver metastasis are one of the two key factors in terms of the Bellman risk criteria, which reduces survival. Overall survival was with a hazard ratio of 0.70 at 10.3 months versus 7.4 months, so there was benefit there. PFS again was not that useful, but combined positive score again starts to differentiate between the two patient groups in that sense as well, in terms of the survival and things like that. So toxicities were similar, well tolerated, so no concerns about that at all, and A's of interest as were quite limited. So it's all coming on quite nicely in that sense. It is survival benefit is maintained, 10.3 versus 7.4 months, so which is important with immune checkpoint inhibitors. The recent EMA announcement is that PDL positivity should be looked at, and again that is based on the phase 1 trial, the phase 3 trial data in first line setting that is going on, and that has been done for both pembrolizumab and atezolizumab in the first line setting. As this is a trial we reported in Lancet recently, looking at the role of ramucirumab with docetaxel, which is a positive trial in terms of progression-free survival, and ESMO will be presenting a little bit more updated data on survival as well. So I think there are ongoing phase 1 trial, phase 3 trials in first-line settings that are going on in platinum-eligible and ineligible patient population group. I think I'll finish with just last few slides on precision medicine. I don't think there's much time to talk too much about it, but what I'm trying to say is that based on this sort of tumor cancer, the Cancer Genome Atlas, and you must have seen the Seth Lerner's paper from Baylor College of Medicine in Cell, October 2017, which very clearly starts to look at the luminal papillary, luminal infiltrated, luminal, and then looking at basal squamous and neuronal subgroup. And that is starting to predict who will benefit more from PD-1, PD-L1, for example, who will be responding to neoadjuvant chemotherapy, and who will have more toxicity, more sort of resistance to neoadjuvant chemotherapy. So a lot of really exciting things coming through. That PD-L1, this is mutational load. It is complementary. It is coming to be complementary to responses. Tumor mutational burden, we talked about high versus low. Starts to, and again, this was in our paper in Lancet, where you saw that although it was looking negative, but if you do a TMB, the TMB positive patients 11.3 months versus 8 months, so there was significantly improved hazard ratio and survival benefit. So it is, as I said about TMB, can start to differentiate between these two different population groups versus low TMB, you see no difference between chemotherapy versus atezolizumab. Then CD8 infiltration is coming into play as well. So there are lots of biomarkers, predictive biomarkers. Maybe in the next months to years we will see them coming into clinical trials and proving themselves and moving forward. And this was the thing that I was talking about, the subclassification of bladder cancer, subtypes of bladder cancer that will help you to predict which patients will benefit and help you to select. And that will be something that you can, when you treat these patients, make sure you've got the tissues to look at them in future, to go through these sort of things as well, to have your own classification, to see what is the difference when we look at Caucasian patient population, for example, versus Asian patient population. And those sort of things will be a very positive addition to future of bladder cancer management. And I will be delighted to work with you all to try and move those sort of things forward in future. This is where I will finish. That is very close to my heart, that it is this sort of variability among individuals that make medicine more as an art, between art and science. Because I think once we start, because the key thing that when you go for assessment and when you go to the reimbursement, the key thing will be these drugs will become so much more effective when you say that biomarker. Again, I'm not saying PDL positive biomarker, I'm saying biomarker positive, which you will define in the next 6 to 12 months, or hopefully in a year or two maximum. You will say that based on that, this has got a response rate where the durability of response will be much longer in those 50% of patients rather than 20 or 30% of patients. And that is what is going to change people's view on reimbursement. Because if you can be so positive that actually it's not like 1 in 4, but I'm telling you it will be 1 in 2 patients who will be long-term responders. That is something we are hoping to achieve in coming years. So that's where I will stop for my first talk. Anik has already told me that I'm running a couple of minutes over now, so I will move on to the next